Okay, let's go ahead and get Friday started. So just a brief announcement. This is not someone I'm hosting, but since this is a microbiology class, and hopefully you're all now even more interested in microbiology than ever, today's seminar at 4 o'clock in the new ISC3 may be of interest to you on the microbiome and impacts on health. So you should actually be making a practice of going to these seminars if you're not already. Okay, and now just a word about so everyone is in the middle of their lab unknowns. Actually, one person has finished their unknowns already. I think it's a new record. But one of the things I want to make sure people understand is that there's a refrigerator in that back prep lab, not just the refrigerator where you're storing your bacteria. There's one in the prep lab that you're welcome to go into. So that back prep lab is not like off limits to students. The TAs like to make you think it is, but it's not. Because they actually sometimes study in there, so they like to keep the rest of you out. But there's a refrigerator in there that if you open it, it's just like home, right? Dig around until you find something good. The only problem is that you can't eat it this time. But all of the, the media that hopefully you might need, the DNase plates, the coagulase tests, things like that, hopefully you'll find in there. And hopefully the TAs or myself will be able to help you find it. So just so you know where to go for resources. Okay, and so since I screwed up my PowerPoint last time and I accidentally transposed one of the slides on generalized transduction, today's lecture is going to be even more disjointed than normal. So I'm going to go just through this one slide on generalized transduction, even though we've finished talking about transduction. But I wanted, for just completeness sake, to talk about this. So as opposed to the specialized transduction that we spent a lot of time talking about, where the only DNA that was going to be transferred between bacteria was immediately adjacent to the site where the prophage inserted, in generalized transduction, any piece of that infected bacterium is capable of being transferred by means of a viral particle. So it's really it's a simpler concept. So here on the top you see a standard infection. A virus has injected its DNA into the cytoplasm of the cell. And in the process of killing that cell, in the process of producing new baby viruses, the host genome gets degraded, in part as resource, to free up nucleotides to synthesize the new viral genomes. But some of those fragments are just broken up into random fragments. Most of what's going to be packaged and released from that infected bacterium is going to be bacteriophage particles that have viral genomes in them, perfect viral genomes and nothing else. But in some viruses, there is just by a random chance, a piece of the bacterial chromosome, the host chromosome, can be packaged by accident. And so that particle, shown here with the designation of a transducing particle, now is, I guess I would say, pseudo-infectious. When that gets released from this bacterium, it has the ability to still bind to another bacterial cell, if that bacterial cell has the appropriate receptor, and to inject its payload, its DNA, into the cytoplasm of that cell. But now that cell isn't truly infected because it's not going to produce new viruses. It's not getting viral DNA. Instead, it's gotten a piece of DNA from the previous host that was killed. And so that piece of DNA, especially if it has homology somewhere in the chromosome, can cross over and produce a stable gene transfer. Probably more often than not, the situation is shown here on the right, an unsuccessful gene transfer where that DNA is recognized as foreign. It's restricted. In other words, restriction enzymes chew it up. But this stable gene transfer is a very important means for transfer of DNA in bacteria. So this is almost always the result of, or it is always the result of the lytic form of the infection. So the bacteriophage has to be able to lyse the bacteria in order to randomly, occasionally take up a foreign piece of DNA. Now, in terms of the viruses, the bacteriophage that can and cannot do this, it actually points out a nice dichotomy in terms of how bacterial viruses package their DNA. There are two basic strategies by which virtually all bacteriophage put, hopefully, from their point of view, their own DNA into the virion and not a piece of bacterial DNA. So they use a strategy either known as pack sites or packaging sites. These are specific DNA sequences that are unique to the viral genome. And the virus has the ability to interact with DNA only if that DNA has that specific DNA sequence, these PAC sites. This is very much akin to, when you look back in your notes, we talked about the Haemophilus influenzae having the ability to recognize an 11 base pair sequence of extracellular DNA and only incorporate that DNA. This is a very similar mechanism. Phage that use PAC sites pretty much uniformly, almost without exception, package only their viral DNA. Viruses that use this strategy are not generalized transducing vectors. They're not very good at picking up foreign DNA. However, some viruses use what's known as a headful strategy. The headful strategy means they will just take any piece of DNA as long as it's the right size. So if a piece of bacterial host DNA, just by random chance, happens to be, let's say, within 5% plus or minus of the optimal size for that head, it gets packaged. So more often than not, what's going to be packaged, because it's going to be the exact right size, is the viral genome. But on rare occasions, if the bacterial DNA that's broken down is the right size, it can get packaged as well. So this is what's known as generalized transduction. So this is another important means for transfer of bacterial DNA. Okay, now for something completely different. Now we're going back to where I left off in the last lecture. I know, even more disorganized than normal, but it's Friday, so just bear with me. So we left off in the last lecture talking about mobile elements. So we were talking about insertion sequences and transposons. So I just want to finish up talking about transposons. So this are just some characteristics of insertion sequences and transposons in tabular form. And so again, you don't need to know the specific details here, but just in terms of a general appreciation, in insertion sequences here, these are 5 insertion sequences that are known in the genome of E. coli. These insertion sequences would be completely different in another bacterial species. You'll see that the lengths go from anywhere from about 800 base pairs to about 1,200 base pairs, which is much smaller, as you can imagine, than the lengths in transposons. So these are various transposons that have been discovered in a number of different bacteria. So the insertion sequences, remember, these are the very simple genetic parasites. They have nothing more than inverted repeats on each end and a transposase gene in the middle. And if you look at this particular suite of them, these are the copy numbers in E. coli. And so some of these are actually present in the F plasmid. So that's what's driving that episomal nature of F plasmids, is shared insertion sequences between the chromosome of E. coli and the F plasmid. That's what allows the F plasmid to go in and out of the chromosome. So that's, in essence, what makes insertion sequences most interesting, to me anyway. Much more interesting, much more impactful are the transposons. Not simply because they're larger, but because they have additional genetic material in them. So if you look at some of the most famous transposons shown here from different bacteria, you'll see that they have acquired some pretty important resistance mechanisms or toxic mechanisms. Things as simple as ampicillin resistance, mercury resistance. One has even picked up the entire lactose operon, so you can transfer to a new bacterium in one fell swoop the entire lac operon. And some of them actually have virulence factors. So the heat-stable enterotoxin that is on E. coli, particularly the E. coli that cause traveler's diarrhea. So these are the things that you try to avoid when you're on vacation, and why you always drink bottled water and you always eat well-cooked food and you don't eat salads, because you're trying to avoid E. coli strains which have this transposon on it, TN1681, which encodes a toxin called the heat-stable enterotoxin, which is what causes the nasty diarrhea that's associated with traveler's diarrhea. So these are really important genetic parasites of bacteria. And if you look, you can also tell that they vary in their complexity. So if you look here at the terminal module, I remember in the cartoon that I showed you that some, but not all, transposons have insertion sequences, intact insertion sequences on each end. So that's what's shown by these last 6. In those particular transposons, on each end of the transposon, there is an IS element called IS50. Or on this one, there's there's copies of IS1. So this would be these would be composite transposons. As opposed to the transposons like TN3 and TN501, these don't have terminal modules. They have inverted repeats, and they have a transposase, and perhaps a resolvase, but they don't have full-blown insertion sequences on each end. My major point here is not that you need to know the difference between TN3 and TN1681. I don't want you to waste your time knowing that. What I'm just trying to give you an appreciation for is the amount of genetic diversity that exists among these transposons. They come in different flavors and different complexity levels. And one of the hallmarks of transposons that kind of makes them easy to spot when you're looking at DNA sequence is, as we mentioned, that they tend to duplicate their target sequence. So here, let's go through an example of a transposon or an insertion sequence going into a new or novel site in a plasmid or in a chromosome. So here is a target site for a particular transposon or an insertion sequence. The transposase enzyme from that insertion sequence or transposon recognizes the inverted repeats on the transposon and recognizes this target sequence. It cuts that sequence, but it cuts the sequence asymmetrically. So here it's cutting between this A and T on the top strand and between this G and T on the bottom strand. So what's left behind are staggered ends, or sometimes referred to as sticky ends. So this is being cut by the transposase, but at the same time, the transposase is taking out the transposon, the genetic element, from somewhere else, or perhaps a copy of it, and it's going to insert it in that cut site. Now, look at how, so this pink bar is meant to represent the entire length of the transposon. It's put in there as a double-stranded piece of DNA. So, remembering the anti-complementarity rule, this C would be the 5', would be on the 5' side. So as we go in this direction, here is a free 3' end on the top of this transposon. That serves as a primer. Remember how DNA polymerases add nucleotides to DNA. They add 5' to 3'. So when there is a free 3' hydroxyl, as there would be on this 3' nucleotide, DNA polymerase can now add DNA, base pairing it with this half of the sticky end, or that staggered end. And so what it does is it generates double-stranded sequence. And it does the same thing from the bottom strand. This would be the 3' hydroxyl end of that transposon being put in, which could now serve as a DNA polymerase primer to synthesize the sequence complementary to this top strand. So you see then that what results, this is the mechanism whereby direct repeats are generated. So here's the target sequence ATAGC on the same strand, in the same direction, 5' to 3', ATAGC. This is a direct repeat. It's in the same orientation and it's on the same strand. This cartoon is not showing the inverted repeats. The inverted repeats would be just internal to that. So the first portion of this transposon here would be an inverted repeat of the just inside end of this transposon. But remember that inverted repeats are on the opposite strands, so you have to look for them differently. So guarantee you will see a question on an exam where you will look for a direct repeat on the outside of an inverted repeat, and as many times as I harp on it, people will still miss this question, even though I tell you that it's going to be on there. But it's a really interesting DNA dynamic that these parasites use in order to make copies of themselves or spread copies of themselves. Now, in terms of how they move from site to site, transposons have two basic forms of transposition, either conservative or replicative. Conservative is the simplest one to think of, and it's the simplest mechanistically, but it's probably the most rare, because in conservative transposition, the element, the transposon or the insertion sequence, is literally picked up from one spot in the DNA and move to another spot. It might be moving from a plasmid to a chromosome or moving from a chromosome to a plasmid. But the original place where it was is left empty. This really is not a good mechanism if you want in terms of genetic fitness, because you're not increasing your copy number by doing that. I think if you're putting yourself onto a plasmid from a chromosome, then the chromosome increases in copy number. There's probably evolutionary benefit to the insertion sequence or transposon there. But conservative replication, from an evolutionary point of view, is, and from abundance, from what we know about how these things tend to work in the real world, this is the more unusual form of replication, even though mechanistically it's simpler. Replicative transposition is horrendously, people spend their entire careers studying how replicative transposition works. But this is probably the more common form of how transposases and transposons move. The element, the original element, remains in its original site, but in the process of transposition, it is copied. So there is an exact DNA copy made at the same time that it's transferred into a new site. So the copy number increases. This makes more sense from an evolutionary point of view because from a fitness of this genetic parasite, it's actually reproduced itself. It's actually increased its copy number. This is, so let's just walk through without getting lost in the mechanistics of replicative transposition. And you can translate that as, and you would be completely correct to translate that as, your professor does not know how replicative transposition works. It's horrifically complicated, and I didn't want to take the time to learn how to explain it. So we're going to go through it in the most generally broad brush strokes of replicative transposition. But here in this particular cartoon, you see an example of, let's call this blue circle a chromosome that has a target sequence that the transposon shown here in orange on this, let's call it a plasmid, recognizes as a target sequence. So the transposase encoded on this transposon has the ability to recognize and to cut that target sequence, as well as recognize its own inverted repeat. And it will make a cut in the inverted repeat. Actually, more appropriately, it makes a nick in the inverted repeat. It breaks one strand and then has the ability to ligate that break from one strand to another on that chromosome. Then DNA polymerase will fill in the single-stranded regions, and what's formed is what's known as a co-integrate. So there is an intermediate form that occurs in the cases of replicative transposition, whereby the entire plasmid that contained that transposon in this intermediate form is completely subsumed. It's completely a part of that chromosome. So now that chromosome has gone from a big circle to a bigger circle, because now it has within it that entire plasmid, not just the transposon, but the entire plasmid. Now there's another gene inside of these replicating or transposing transposase. Yeah, I really messed that sentence up. There is another critical enzyme that's present, that must be present in transposons that utilize replicative transposition. And that enzyme is called resolvase. Resolvase, beautiful name, because what it does is it resolves the co-integrate. After this co-integrate is formed, resolvase will take the original plasmid, complete with the original copy of the plasmid, back out of that chromosome. So it backs out. Much like the F plasmid has the ability to remove itself at some frequency from the bacterial chromosome, making it an episome. But in this particular case, resolvase is taking that co-integrate, which is only an intermediate form. It's short-lived. And what's left behind in the chromosome is nothing more than the copy of that transposon. The rest of the plasmid has been removed, and the only thing that has been moved in is an exact DNA copy of that replicative transposon. It has duplicated the target site, and then just inside of those duplicated direct repeat target sites, you'll find the inverted repeat and all of the rest of the genetic material of that transposon. So this is replicative transposition in the most simple way possible. This cartoon actually, I think, maybe shows it a little more clearly. So here you have a plasmid that has E. coli's TN3 transposon in it. This is a replicative transposon. So this transposon contains a transposase and a resolvase. It has to in order to do this. Here is the target plasmid, which has a sequence in it that the transposase can recognize. So the transposase of TN3 recognizes its own internal repeats and has the ability to nick that DNA within those inverted repeats. It also has the ability to cut that plasmid at that target site on each strand, generating those staggered ends, and to form a co-integrate, where now you have two copies of TN3, the original one from the plasmid, and now the one that was generated by replication during this integration step. But you have all of plasmid A inside of that cell too. So you can see that this co-integrate is much larger. But this is a short-lived intermediate structure. It doesn't stay for very long because the enzyme resolvase is now going to use what are known as IRS sites or internal resolution sites. These are DNA sequences within the transposon that it helps to mediate this kind of a site-specific recombination, which is going to regenerate the original plasmid. It's going to resurrect it from that co-integrate exactly as it went in. Not a base pair change in that. It still has a transposon which can do this same process again and again. If this plasmid is a conjugative plasmid, then it has the possibility of being passed into a whole new bacterial cell, and so that whole new bacterial cell might be a target for TN3. But you see that the chromosome now is modified only in having an additional copy of TN3. So replicative transposition increases the copy number by 1 every time a replication event occurs, or each time a transposition event occurs. Now, one of the other reasons why it's important to understand these things, other than the tremendous interest in just basic science of bacterial genetics and the fact that transposons often carry antibiotic resistance, people have actually done a really clever study where people noticed that the integrase of human immunodeficiency virus is actually structurally very similar to the transposase of TN5 from E. coli. So if you look at the alpha helices, beta sheets, and the catalytic residues, there's obviously some similarities between that. So what a clever group of people did was they screened transposase 5. They screened a series of drugs or chemicals and found that a large number of the small molecules that interfered with E. coli transposon 5's transposase also interact with HIV-1 integrase. So some of these drugs may actually make it into our chemotherapeutic ability to treat people infected by HIV. So just another reason why it's important to understand transposase. Okay, we're done with transposons. Let's move on and talk about a really weird genetic element that I really didn't know where to put in this class. So I'm putting it in with mobile elements, even though itself, it is not mobile. So this is a DNA structure called an integron. We're going to link it with mobile DNA because it has the ability to mobilize DNA. Itself is by itself, it's largely sedentary. It's in a bacterial chromosome, and for the most part, it doesn't move. I think there are some examples of integrons that have been found in transposons, so they may be being moved passively, not under their own control. But since they have the ability to move DNA into themselves to acquire foreign DNA, we're going to kind of classify them as a mobile DNA. They have some similarities in terms of plasmids. They're related to transposons. I meant to say transposons. They're related to transposons, but they're not flanked by direct repeats. So this is a kind of a basic anatomy. This is probably the simplest form of an integron that's been found. It's called IN0, so capital I, lowercase n0. So this either means, and I'm not sure which is the case, it either means it was the first integron discovered or it's the simplest integron discovered. I suspect it's the simplest integron discovered. But it makes for a good study of the anatomy of this piece of DNA. So integrons always have an integrase gene. So this is kind of similar to transposase. This is a gene whose gene product, whose enzyme has the ability to integrate foreign DNA into itself, in essence. That's what this capture sequence is. So the enzyme product of this gene int1 has the ability to take foreign DNA, if it has a specific tagged sequence to it, and insert it at this capture sequence, shown in green. That capture sequence is not coincidentally located just downstream of a promoter. That's what that red bent arrow means. Whenever you see a bent arrow like that, that means the start of transcription. So it implies that there's a promoter there. So by putting a foreign piece of DNA there, it's basically putting it under the control of a promoter that already exists. So the bacterium now has the ability to express this new piece of DNA that the integron has just slid into that capture site. So in integron 0, IN0, there are two genes present. One gives resistance to ethidium bromide. I don't really know what the benefit to bacteria is. I always just think of ethidium bromide as something we use in the lab to make DNA fluorescent in agarose gel. But there is a gene that gives bacteria the resistance to ethidium bromide. But there's also a gene in there that gives bacteria resistance to sulfonamides, a type of antimicrobial. So you can see that having these genes in here, especially the resistance to sulfonamide, could help a bacterium survive treatment with that particular drug. But the integron is not happy at that point. It wants to grow, and it does grow tremendously. So it can capture foreign DNA and insert it at that at site. So in integron 7, you can see that it's added this AADB gene. AADB is a resistance gene for aminoglycoside antibiotics, things like tobramycin, gentamicin. So some pretty important antibiotics that are used clinically. This is a gene that gives bacteria resistance to those aminoglycosides. Other forms of integron have not only incorporated AAD-A, but put in OXA2. OXA2 is actually an antibiotic resistance gene as well. It gives bacteria resistance to beta-lactams. It's another way to be resistant, so it's a degradative enzyme that breaks down beta-lactams. So you can see that over time, by acquiring a foreign piece of DNA like this, and these are kind of mysterious pieces of DNA in terms of where they come from originally. Because in the laboratory, we know that we can do this, and the DNA tends to always be in a circular form and have a particular sequence called ATC, which is somewhat related to the 59 base pair capture site called ATI. So ATI meaning attachment to the integron. ATC, C standing for cassette. So these are referred to as gene cassettes that are going to be inserted. So the integrase gene product, the product of this INT1 gene, actually does the site-specific recombination between the ATC site of this new piece of DNA that it's about to insert and the ATI site. And what it does is it ends up putting that new open reading frame, shown here as ORF-X, directly behind this promoter. So now ORF-X is going to be expressed. And if there's a good phenotype associated with that, if it gives resistance to an antibiotic, the bacterium has just acquired resistance to that antibiotic. So these integrons capture novel DNA. Now, I'm rethinking this, especially antibiotic resistance genes. That's probably not entirely true. We have so integrons were discovered because we're interested in how bacteria become resistant to antibiotics. So of course, most of the integrons that we've discovered in the microbial world tend to have antimicrobial activity associated with them. But there's a natural bias. That's what we're out there looking for. That's what we're interested in. I will bet in nature, once we reach a point where we've looked at every integron in nature, we'll never actually do that, but we'll find that the acquisition of genes is not limited to antibiotic resistance. But that's what we know most about. So this novel DNA that's coming in probably is a very rare event. We don't really know where this DNA is coming from because, it's double-stranded. So DNA is not generally taken up very frequently as double-stranded DNA, and it becomes circularized. So where these genes originally came from, at least in my mind, is very mysterious. What's perhaps more interesting, we'll get to in another slide, is that all of these genes that have been acquired over the millennia, so this has probably occurred over millions of years, are actually going to be shuffled at some point during bacterial growth, during times of bacterial stress. So it's not a matter of most times what's being put in at this capture site is probably not a new piece of DNA that came in from the environment. It's probably a piece of DNA that has worked its way down the line. So this SUL1 gene that gives sulfonamide resistance, over time, over thousands of years, as new pieces of DNA have been put in here, it's been pushed to the end of the road. It's perhaps not being expressed at a very high level because maybe one of the genes ahead of it has got a transcriptional terminator. So this gene is kind of an orphan. It's kind of lonely down at the end of the line. Well, what I'll show you in a minute is that under stressful conditions, what bacteria can do is they can shuffle all of these genes, kind of like generating random numbers or picking a lottery number, if you will. That these pieces of DNA can be excised and circularized and perhaps by random chance moved into this site. So once the genes are in here, they're a great resource just waiting to be used. Yes?
Does it always maintain frames?
I'm sorry, does it always what?
Maintain frames? So it doesn't have exact frames that are being started with.
Right, so does it maintain frame? No, probably not. But it wouldn't really be important because each one of these genes, even down here, that would be transcribed on the operon would still have its own Shine-Dalgarno sequence. So it would bind to an individual ribosome each time. So the frame is not so important. What I think, but it's a good point, because I think what's more important is the fact that, let's say here, when OXA2 was put in, this sequence at the end of OXA2 might contain a transcriptional terminator, an intrinsic terminator, which would stop the transcription, so that mRNA would stop here, which would mean that the subsequent genes might not be transcribed. And actually, even in the absence of a transcriptional terminator, just by attrition, RNA polymerase may tend to drop off. So the further down the line a gene is, the less likely it is to be transcribed, or it's going to be transcribed at lower and lower levels. And in fact, a lot of these genes become pretty much silent, and so they are subject to genetic drift. The mutations occur in them with no selection against them. They accumulate that way. So the integrons are usually sedentary, but some are on mobile elements. One, in particular, is really important for medical reasons, but it's also just like the biggest integron ever. It's actually been referred to as a super integron, although I think people are getting away from that term super integron. But Vibrio cholerae, the bacteria that causes epidemic cholera, which is sadly raising hell in Haiti right now, has 179 different genes inserted behind it. It's more than 3% of the total genome is a part of this super integron of Vibrio cholerae. I like this slide better because it shows better, I think, how these rearrangements can occur. So again, we're back to the SOS response. You thought you could forget about it. I know you're studying for it now for exam 2, but it keeps coming back. This will illustrate why it's so important to understand about the SOS response. If you drill down and look at the genetic locus here for the INT1 protein gene, as well as the promoter that's going to drive the transcription of these cassette genes. The promoter that drives the transcription of INT1, the integrase, is actually part of the LEX-A regulon. It's part of the SOS response. This is wonderfully evil. The bacteria has evolved the ability to put this integron, which contains a whole number, in the case of Vibrio, 179 different genes, that it has the capability to randomly reassort under SOS-inducing conditions. And SOS-inducing conditions are not, as we talked about, simply DNA damage due to ultraviolet light or something like that. Antibiotics that damage DNA, such as ciprofloxacin, one of the most commonly used broad-spectrum antibiotics in use today, as well as beta-lactams, actually can induce the SOS response. And so what happens when the SOS response is induced is that LEX-A is going to be degraded. And so you're going to get an increased transcription in the integrase. You're going to get also an increased expression from this PC. These should be lowercase, promoter, subcase C, meaning the promoter for the cassette. C here stands for cassette. This word is not pint. The INT should be in subscript. This is the promoter of the integrase gene. So you get an increased transcription in the case of an SOS-induced response of the integrase, increased expression of these genes, but you also get all of these other genes down here at some frequency become excised and turned into circular DNA. So that in this particular case, this cassette down here, which has probably been undergoing neutral genetic drift because it hasn't been expressed for perhaps thousands of years. So maybe it has a function now that's novel, or maybe it still has its original function. But at some frequency, each one of these genes down here has the ability to be excised, circularized, and then reinserted at the head of the line. So this gene, which hasn't been expressed for, let's say 2 million years, now finds its way here under the control of the promoter for the cassette, and now that bacterium can express that protein. If that protein helps the bacterium survive the stress, whatever it was that induced the SOS response, great. That bacterium lives. Chances are it still dies from the stress. But with 179 different genes to choose from, if one of those genes has a benefit to help the bacterium survive that stressful environment, then that one cell is going to survive and so is going to reproduce. And so now you have a change in the population. So you've changed the order of these genes. So you brought one up to the front because it was beneficial. And if the stress occurs again in another 10,000 years, maybe a gene further down by random chance gets moved up in here. So it's very much like a lottery. So there's no guarantee of a winner. It's not certain that one of the genes downstream in that cassette can actually overcome that stress. But hey, if the alternative is the entire population is going to die due to stress, then why not roll the dice and just completely shuffle up within the population this genetic element? VR, did you have a question? So does that, so if you moved, so VR's question is if you just moved a gene down here randomly up into here, doesn't it interfere with this gene? So you're talking about at the protein level or at the DNA level? I mean, it is possible that the gene product of the new gene that's been inserted at the head of the line here could positively or negatively interact with the gene product from one of these other genes. That's certainly possible. I think what's more likely is that it potentially interferes or does something to the transcription of those genes. So I think it would be a very random event to have the two proteins interact with one another, but it's certainly possible. It's the great thing about microbiology. Numbers are so huge that virtually anything, any rare event is absolutely a certainty. I mean, somebody's going to win the lottery, right? You know, it's not going to be you or me, but somebody always wins the lottery eventually. You know, it's just bacteria are the same way. They're so small, their numbers are so big, eventually by just random chance, somebody wins. Now, if you look at, so people have actually done the studies in Vibrio because Vibrio is, of course, of great interest to people and it has the largest known integron. SOS induction of Vibrio cholerae in the lab increases the expression of that integrase gene 37-fold. So getting rid of the SOS or Rec A, degrading that Lex A on top of the int promoter, has a tremendous upregulation of the expression of that gene. And there's a 340-fold increase in the cassette recombination. So if you take in the laboratory and just stress this bacterium out and then look at the results later, you'll find this, yeah, I mean, I keep coming back to my metaphor of the lottery, this rearrangement, randomly throughout the population. And hopefully somebody is more fit after that arrangement. Okay, that's the end of that one. Now, this is going to sound weird, but now it's time to start microbiology. We're actually now going to start talking about microbes in the real world, so how they interact. So we've gone through a lot of the structural stuff, we've gone through a lot of the chemotaxis and the genetics. Now let's talk about microbes and their diversity and go through some real life stories of how microbes exist in the environment and what they do. Really what we're going to do is, so I'm not a phylogeneticist, so those of you who are worried about phylogenetic trees, don't worry about it. But what I want to do is to spend a little time talking, not so much about phylogenetics, but actually I'm looking to get a tattoo. And so as I look through online, this is the tattoo that I think I want to get. I don't know so much about the badger. I think I want more bacteria on here or some more birds. But, I'm not going to get a tattoo. But if I was, this is a cool one. And I thank the class member who let me take a picture of. No, I'm just kidding. Okay, so we're not going to do a lot about phylogenetics, but we have referred periodically through class to this mysterious domain of life called the archaea. And so I want to spend a little time dedicated to the archaea. So this modern tree of life really came about during my scientific career, because when I was, growing up in science, archaea were referred to as archaebacteria. We thought they were just weird bacteria. There was a guy, Carl Woese, University of Illinois. One of the greatest tragedies of Nobel Prize being overlooked in history. And that sadly, the Nobel Prize can never be awarded posthumously, and Carl Woese died a few years ago. But this guy labored in anonymity in a basement lab, no windows, at the University of Illinois, Urbana-Champaign. Brilliant microbiologist. And he kept, this was back when DNA sequencing and RNA sequencing was not very easy. But he kept manually sequencing the 16S RNA of these weird archaebacteria. And it took him a while to convince people, but he really changed the whole paradigm of the domains of life, because he was very easily able to show, once people took him more seriously, that these archaebacteria were not really bacteria, and they were not eukaryotes. They were somewhere in between. So in a way, I hate the term prokaryote. It's like I have a love-hate relationship with the word prokaryote. I love it, but in a way I hate it because it's a very artificial term. It's a man-made term, just meaning microbes that don't have nuclei, they don't have intracellular organelles. You can tick off all of the trivial pursuit answers to the difference between prokaryotes and eukaryotes. But prokaryotes really are archaea and bacteria, but that tends to make people think that archaea are more closely related to bacteria than they are to eukaryotes, because they're both in this collective term prokaryotes. And it's not true. Archaea are no more closely related to bacteria than they are to eukarya. So again, don't worry about the details in a table like this. This is the kind of table that puts you to sleep when you go through a textbook. But this is a table that shows the differences and similarities between bacteria, archaea, and eukarya. And so don't worry so much about the words on here. But what I did was I added on top of that kind of a color coding. So I put a red cross or star in front of characteristics where archaea are similar to bacteria, and blue stars in front of places where archaea are similar to eukaryotes. So if you just kind of look at this without paying much attention, you can see almost an equal amount of blue and an equal amount of red, and only a little bit of green. So the green arrows indicate unique archaeal properties. We've already talked about one of them. Archaea are unique in having that ether-linked and branched aliphatic chains, the weird membranes that they have, and the lipid monolayers. So you already know about one of the unique aspects of archaea that's found in no other domain. You also, I think I mentioned, but we'll talk a lot more about it, remember that the only organisms on Earth that can biologically produce methane are archaea. So these are only the two things where archaea are different than bacteria and eukaryotes. But look at how similar they are in many cases to eukaryotes, to you and I, and then to bacteria. So they're clearly something unto themselves. The DNA-dependent RNA polymerase that archaea have is clearly much more related to us than to bacteria. Some of the proteins, so people drawing these cartoons always have an agenda, right? So if you make two things the same color, of course you think they're related to one another. But in reality, at least two of the proteins in the archaeal RNA polymerase have direct eukaryotic homologs in RNA polymerase in eukaryotic cells. So it's clear that we share a lot more transcriptionally. And actually, that's borne out by the fact that certain antibiotics that we use, such as rifampicin, to treat bacterial infections, rifampicin affects or stops bacterial RNA polymerase, but our RNA polymerase and that of archaea completely unaffected. So again, showing, because RNA polymerase is a good target for antibiotics, and it points out the fact that the RNA polymerase is very different between bacteria and archaea, despite the fact they're both prokaryotes, despite the fact they're both microscopic and require a microscope to see. Archaea also have histones, something that bacteria don't have that we have. So this is the first evolutionary demonstration of histones. It's actually present in the archaea. Again, it's a phylogenetic tree, I know. My eyes glaze over when I see these two, but it's a really simple one, because basically all we really want to talk about are the major phyla present in the archaea. We're going to talk about the Crenarchaeota, the Foam Archaeota very briefly, and the Euryarchaeota. And we may even mention this core or we will mention this core archaeota. Now, of the 36 major phyla of prokaryotes that are known, the majority of which we can't grow in the laboratory. So 36 phyla, only 26 can be grown in the lab. That means that most microbes in the environment, in the world, we don't know anything about. We're only learning about them now because we do something called metagenomic sequencing, where we can sequence DNA from the environment without growing the bacteria. So of the 36 major phyla prokaryotes, 2 are archaea. So the core archaeota are recognized as kind of a separate phylum. And they're found, for the most part, I think, or they were at least found first, in the obsidian pool in Yellowstone. So one of the great things about studying archaea is that you get to live in Montana and work in Yellowstone National Park, because that's where most of the discoveries are going on in archaea, especially the Crenarchaeota and the core archaeota, because they tend to live at high temperatures. So these core archaeota are now recognized as a non-cultivatable phylum, but we still don't know that much about them. The Crenarchaeotes are the ones that are particularly interesting to people who work at Montana State, which is the mecca for people working with extremophile microbes. These are hyperthermophilic organisms that we've talked about before, whose optimal temperatures is often over 100 degrees centigrade. So these are microbes that have been around since the dawn of time, since the dawn of life on Earth, because they're adapted to how life on Earth used to be. And I think rather than continue on with this, I lost track of the time. We're close enough to the end that we'll just stop here and pick up on Monday. Have a great weekend, everybody.